I am Dinesh Kalra from Dayanand Public School, Pandusar Nabha. In this video, I will discuss Meselson and the Stahl experiment from chapter Molecular Basis of Inheritance. DNA replication is the process of formation of the new DNA from old DNA, and it is the basis of inheritance. It can be explained by central dogma, which states that genetic information flows only in the one direction. You can see from this figure, from the DNA, by the replication, new DNA is formed. Then this DNA undergoes a transcription, it will form the messenger RNA. Then messenger RNA undergo the translation and proteins are formed. Proteins will control characteristics of an organism. That is, if there is a change in the DNA, then there will be change in the protein and ultimately it will change the characteristics of an organism. There are three modes of replication: conservative, dispersive and the semi-conservative. According to the conservative mode, after the replication, parental DNA remains together and newly formed DNA, they remains together. Means there is no mixing of the strand. Second is the dispersive mode. So according to this, parental strand and newly formed strand, they will mix together. Third is the semi-conservative mode of DNA replication. In 1953, Watson and Crick proposed this model. According to this model, two parental strands, they will separate. Each strand act as a template, and after the completion, each DNA had one parental strand and one old strand. As you can see in this figure, after the conservative mode of the replications, old strand and new strands, they are not mixed. In the semi-conservative, in these two DNA molecules, one is the old strand, second is the new strand. In dispersive mode of replications, both new strand and old strand, they are mixed together. This is the most acceptable model for the replication. For this, Meselson and the Stahl in 1958 gave experimental proof, and it is the most beautiful experiment in the biology. They used E. coli as the model organisms. Two different isotopes such as N15 and N14 were used in this experiment. When these isotopes were incorporated into the DNA of different density were formed. If in both the strand N15 is incorporated, this is a heavier DNA. If both strand has N14 DNA, then it is a lighter DNA. If one contains N15 and other contain N14, it is a heavy DNA and it has a density in between these two. Regarding the experiment, first of all, they prepared heavy DNA. For this purpose, E. coli was grown in a media which contains N15 isotopes. After some generations, this N15 was incorporated into the bacterial DNA. Then, these bacteria were shifted to light media, that is, in a media which contains lighter isotope N14. After the interval 20 minutes, sample was taken, DNA was isolated, then was subjected to density gradient centrifugation based on cesium chloride. What did they observe? After 20 minutes, extracted DNA showed an intermediate density band, that is, a DNA which is a hybrid contains 15N and the 14N. These findings disapprove the conservative mode of the replication, according to which there should be the two bands. After 40 minutes, two replication cycles, two types of DNA bands were observed. One was of the hybrid DNA and other was of the light DNA. As you can see here, first of all, they grow the bacteria in a media which contains N15 isotope. Then shifted to a media contain and 14 isotope. After 20 minutes, there was only one type of DNA. So it contains only one band. After 40 minutes, two bands were observed. One was of the hybrid DNA, one of the lighter DNA. They have the ratio 1:1. After 60 minutes, the ratio was the 1:3. From this, they concluded that DNA replication is semi-conservative in nature. Each new DNA double helix contains one parental strand and one old newly synthesized strand. Thank you.